We'll start the Q&A session now. So the first question is for Thomas. Have you looked at, it's from Cecilia, have you looked at whether providing pairs of other combinations of fibers results in different short-chain fatty acids production from each fiber in isolation?
Yes, we have. And but only in a very limited manner. It's an interesting question, and actually we're collecting data on that in a way to answer more rigorously, but it does appear that, for example, if you combine inulin and pectin in equal quantities, the resulting production, at least at the doses we consider, are a linear combination of the two. Now, that being said, that's at the dose that we're considering, and I think that we're potentially right in the sweet spot, you know, and there could be some saturation effects or some competition between pathways and so on. So it's a very interesting question, and actually quite a complex one when you get into the nitty-gritty.
Thank you. The next question is for Julie, is from Alex. Is in your artificial gut system, do you see C. diff plume when antibiotics are not administered?
Yeah, no, it's actually not able to be maintained really without the antibiotic treatment. So under those conditions, the gut microbiota is doing, we now know a number of different things to suppress the growth of C. difficile. So it's producing valerate. Some data I didn't get to present, but other groups as well have shown that biometabolism is really important for C. difficile growth. So the gut microbiota also consumes a progerminant that's something called taurocolic acid. And so if you have a healthy gut microbiota, you're also consuming that, and you're also producing another inhibitory bile acid, so deoxycholate. So there's a lot of different ways that a healthy gut microbiota is not supporting the growth of C. difficile. So yeah, we need that antibiotic treatment.
The next question for you, Thomas, is actually a topic of a bunch of the questions that we've received is what drives the inter-individual variability in fiber fermentation capability, and is long-term dietary habits, early life exposures, or other factors altogether? And I'd like to connect it with a few other questions. You talked about that the fermentation capability is quite stable across time. So one of the other questions talked about how stable it is across age. And I was curious how stable it is across, across diets. Like, have you tried to test it at a more finer time scale, like after a meal, after a different type of meal?
Right. I mean, it's a great question, and it's like quite a rabbit hole, that one. I think that working back, we haven't tested long-term dietary interventions or even short-term ones, but it's a very interesting question. We do know, though, that if sampled between several days apart, we get very similar results. So that being said, when I say very similar, I say within the variability that you can expect between 2 different days of a stool microbiome. And so really, I think that the main drivers can be considered, at least at first order approximation, to be similar to the drivers of microbiome composition. It's a sort of functional property of the microbiome, the fermentation capability. And so diet does, of course, contribute to that. Long-term fiber intake will likely improve fermentation capability, just not through any selection pressure or other than simply providing the food for the bacteria that are good at fermenting it. So I think that in that respect, this is a rather simplistic answer, but the answer is yes, the diet seem to be a big driver. And ultimately, I don't think that there's any higher order effects there that I can comment on at this point, namely perhaps some host feedback from, having production of certain metabolites that are of interest to the host, potentially immunity. That's something also, the effect of SCFA production on host immunity and the feedback back to the microbiome composition is something. These are things that are very poorly understood, at least by myself, and so I can't comment on. But I think that overall, the way I think about it is fairly lowbrow, and that it's the same thing that shapes your microbiome composition. So namely diet, lifestyle, environment, and so on.
Thank you. Next question is for Julie, from Namrata. Namrata is saying those are really cool results from Valerate Therapeutic in the CDI model. Is the benefit due to direct inhibition of C. diff, or is the Valerite feeding the beneficial microbes and promoting their growth?
Yeah, so I did the pure batch culture experiments where I just took C. difficile and incubated with Valerate, and I saw it did decrease its growth, but I did see that gut commensal also had its growth increased as well. So it could be a bit of both. Yeah, so we do know it is by itself inhibiting it, but there could be other effects as well. Yeah, that's a good question.
Okay, the next question for you, Thomas. Your data from Namrata again, your data very nicely shows how specific stool samples might be biased towards production of certain short-chain fatty acids, even with the same fiber. But not all short-chain fatty acids are equal in terms of their benefits to the host. Do you want your fiber intervention to remodel the microbiome to correct this bias, or else just capitalize on existing short-chain fatty acid capability?
Well, I think that it's easier to do the latter. So as a first attempt, that's actually very much the angle we're taking, while a lot of people are working on microbial therapeutics consisting of actually trying to modulate the composition and so on. And that's fantastic. That is the future of the field. We're taking a sort of very low tech approach of working with what you currently have and saying, okay, if we can find a good enough links between short-chain fatty acids and a particular indication, then we can say, try and boost butyrate production in a ulcerative colitis patient. And then we take that where we just work with what they've got if they're capable of it. So ultimately, I think that rectifying but the very interesting thing, and this is something that we're collecting through time, and it's going to take some time, a couple of years probably, to get enough data on that. But through time, people taking specific fiber supplements and sampling their fermentation capability through time, we expect that just for the reasons that I was talking about earlier, if you eat the food for the bug is going to grow up, and there'll be more and more of it, and likely then will reflect in your fermentation capability as an even stronger ability to produce that short chain fatty acid from that fiber. So I do expect that we can modulate and change that if that's what the question meant by bias. But certainly that's a sort of a more long-term goal that isn't yet found in evidence. I think what we certainly can do is take a particular patient, and assuming we can link it with short-chain fatty acids, their indication, I mean, boost that production with their existing framework.
I have a question, might be a little funny, but to Julie, what do you think about, like, once you have transferred the poop donation into your platform and to your in vitro system, could you then do some form of like in vitro evolution of this sample to make it less and more resistant to, for example, CDI? And then could you then use this sample to put it back in the same patient and kind of alleviate or mitigate some of the risks for FMT?
As you say, like take someone's fecal sample, do something to it, and then give it back to them so that it helps them fight. I think that's a really interesting idea. I haven't really thought of that. I think, we have thought about taking a stool sample or a defined microbial community from our artificial gut system and then giving it to a patient, but not taking it from the patient to begin with. But I guess there are concerns. You need to make sure that you don't accidentally introduce contaminant, because obviously if you then give that to a patient, that could be bad. But so if you could very precisely control for that, that could be interesting. But then I guess if it gets back into the patient, you might lose whatever selective pressure you had that you were applying in your in vitro model. So you might have to maybe do other things like modify the diet or stuff like that to try to get those, whatever bug or population it might be, to remain increased. But that's interesting. I mean, I guess that's the nice thing about these in vitro models is you have a bit more freedom to do just kind of whatever you want. You don't have to worry about ethics, really. So.
Yeah, another related question, I guess, to you both is a lot of people are trying to actually change the composition, and like you both are focusing on like the metabolic capacity of the specific composition and how it can be changed. And you think like the future treatment would focus on like feeding or underfeeding certain species, or maybe is there a potential for these specific metabolites to actually beat a treatment?
Julie, I don't know if you want to start. I'm happy to. I've got one thing I just want to say that's again fairly lowbrow, is that butyrate smells like vomit. Propionate smells like wet feet, you know. So it's literally, I'm not joking. So it's sort of there's been there is evidence, for example, in ulcerative colitis patients, that if you give them butyrate enemas, that you observe improvements in the disease course, but it's just not a treatment modality that's either patient friendly or even probably appropriate, given that we're dosing the short chain fatty acids extremely high, potentially at levels where we're impacting host physiology in ways we don't understand, as opposed to exploiting the chronic capability of the microbiome to produce it at healthy levels. And so in that respect, I tend to shy away from using the SCFAs themselves as treatment, because we can exploit the bacteria to produce them in a chronic and slow release fashion. But I'll limit that to short chain fatty acids, and maybe that's where Julie might be able to, and short chain fatty acids, specifically butyrate and propionate and acetate. There are others, and maybe Julie might have something else to say about those, I don't know.
I'd add that valerate also smells horrible.
Yeah.
But actually, glycerol trivalerate hardly smells at all, which is interesting. So if you wanted to use glycerol trivalerate as a treatment, it is something that you can actually drink in theory. I've not tried it, but you at least can't smell it. But yeah, I mean, maybe if you're thinking about things like metabolites, it might be useful for the infection point of view. So maybe if you, for C. difficile, the problem is when you stop antibiotics, the C. difficile starts growing and you're not giving them a chance to recover their microbiota because they're just have to go back on antibiotics because they're sick again. But if you're able to suppress the growth of C. difficile and allow their microbiota to recover, and use maybe valerate, for example, as a sort of a temporary treatment that you're dosing in, and maybe once you stop that valerate treatment, once their microbiota has recovered, then you know what I mean? Like you give their own microbiota a chance to come back. So I think they can have applications, but I mean, I can see that if you're thinking of it from a dietary point of view, you're saying, okay, eat butyrate for the rest of your life. That might be more hard for some people, but maybe the companies might like that more because they can sell it. I don't know. But yeah, so I think it kind of depends on what you're looking at. But I think there's sort of different perspectives you can take on the idea of metabolites as therapies, or bugs that produce metabolites as therapies.
Okay, next question for Thomas is from Sean Gibbons. Are these fibers consumed uniformly across patients, independent of short-chain fatty acid production? Any evidence that gas, for example, CO2 production, is higher in folks who are less efficient at producing short-chain fatty acids?
Mm-hmm. Yeah, I mean, it's a great question. We have limited data on that question, but in vivo, it seems that the only fiber that we tested in terms of it being consumed was inulin, and it was in a very accessible form and a particular degree of polymerization that made it such that we were almost barely detecting it in the stool after ingesting 10 grams a day. Now, when we look in vitro, we do see, I don't have the statistical strength behind our data to say this with full confidence, but that there seems to be a clear relationship between the produced total SCFA load and the amount of fiber fermented. And ultimately, those two are separated by some intermediates, but assuming those intermediates move along the chain, one converts into the other ultimately. So there should be a fairly direct link if you're able to ferment it. However, when it comes to the question of gas, so that's another really interesting and difficult question that I think no one knows for sure. However, I will say that all evidence points to the fact that producing gas is indicative of fermentation, which ultimately results in short-chain fatty acids. Also, the interesting thing is that the stoichiometry seems to suggest that different fibers will result in different amounts of gas. And indeed, we see that. It's actually very clear in vitro that inulin is a very gas-inducing fiber. I think people who've ever tried consuming inulin, at least a fraction of us will be able to say that indeed that's true. And indeed that shows in vitro too. About 30% of people produce bubbles in vitro when you feed them with inulin. And that is related to actually a high production of total SCFAs in the medium. So all evidence seems to be that they're linked very much, but not directly necessarily, because it's specific to the fiber, I guess is what I mean. Pectin doesn't necessarily result in as much CO2 as inulin.
Thank you. Another question to Julie from Mario. Is there any information about the transcriptional response of CDs to Valorant?
No, not to my knowledge. So it's not something we've done yet, but could be something that we look at in the future. So I mean, I don't know anyone else who's looked at, I know some people who've looked at measuring valerate from stool from patients, but I don't know anyone else who's really looked at it sort of in a lab setting. But that's not something we've looked at the moment, no.
Thank you. Thomas, Sean is hitting again. Back to the diet question. I know that you enforced a similar dietary background across participants in your feeding study. Can you talk more about how that influenced the results?
Yeah, it introduced all sorts of artifacts that complicated our analysis instead of simplifying it. One problem is that the milkshake we used, Ensure, is very low in fiber, very high in processed sugars. Actually, it contains little else. It's mostly dextrin. So the result is that you're providing a very ubiquitous substrate to the microbiome. And what we saw, that's the hypothesis at least, is the ubiquity of the substrate, is that we saw greater stochasticity rather than in reduced variance between participants. In fact, some of that, if you recall, during the talk, I briefly talked about the strain level diversity in response to the diet. And what we found systematically was that strain diversity increased after 3 days of the standard diet, not only inside an individual, but between individuals. So it seemed to actually throw everything off have the opposite effect. And I think that was, rather than the choice of standardizing the diet, rather a poor choice of what we use to standardize diet. And indeed, that seems to be consistent with the fact that the signals that we find in feeding studies across the literature tend to come from fibers and ingredients like that. There are other signals too, but fibers are more reproducible than anything else, whereas feeding glucose to a medium is likely to introduce a bit of stochasticity. So that was one thing. The other thing I'll note is that huge amount of sugar, or I don't know what it was, but what we found was that the microbiome appeared to be quite stressed. We saw a great increase in the relative abundance of different phages, which were indicating that likely they were entering the lytic phase of their life cycle, and in response to stress. We also found much greater strain diversity across different organisms, and this across the whole tree of life, not phylogenetically constrained, which some evidence points towards the fact that the fitness landscape flattens when under stress, when a system's under stress. So overall, the effects did not seem to be good. And in hindsight, that was actually quite a mistake. But it was interesting nonetheless, and I think that people should stop giving Ensure in the clinic. That's all I'd say.
Thank you, Thomas. The two next questions I'll join together for you, Julie, is about the culture, the conditions of the culture. So the first question from Priyanka is the culture always in an exponential growth phase in the artificial gut setup, or have you observed the culture undergo dormancy? And then the second question is the culture medium provided at a more or less constant rate? And similarly, what is the rate of removal?
Good question. So the first question, I think they're generally considered to be in stationary phase. So not exponentially growing, but not dying. So yeah, stationary phase is the short answer for that. The next part of the question was about the addition of the media. What's the rate? Is that the question? Okay. So yeah, it's added it's constantly added throughout the entire experiment at the same rate. And the way it's set up is that once the level of the culture starts to rise, once basically it's a pressurized vessel. So once there's a little tube that kind of dips down like this, and once the level of the culture rises above that tube, it gets sucked out of the tube. So it's always maintained at that set level. So the rate of addition and the rate of expulsion are equal, so that you're not, because otherwise you're going to have the volume of the culture. Even if you have two pumps that are pumping at the same rate, it's not going to be the exact same, and you can have the total volume vary. So the way that we run them, the volume is constant and the nutrient addition rate is constant, and that helps to maintain the stability of the community. So yeah, I hope that answers that question.
Certainly.
Yeah, should have had a video for that, but yeah.
Thank you. So Thomas, another Tom, has a bit of an odd question. Do individual dietary preferences show any correlation with our ability to ferment specific fibers? In other words, do we tend to eat fibers that are more readily fermented by our unique gut microbiota?
God, that's odd but very cool question. I think that there's no evidence of that, but I love the question because there's plenty of reason to believe that we might over time, given that butyrate, for example, in large-scale cohorts where they try and correlate different metabolites with different lifestyle factors, butyrate seems to correlate most strongly with positive things and high quality of life scores in terms of just general wellness. And so it's only a step further to suggest that perhaps over time you would naturally, unconsciously be drawn to particular fibers that result in favorable short-chain fatty acid production. And so that could be some pretty interesting way by which these bacteria are like favoring their own selection. That's it's a high level effect that I think would be, I don't even know if it's that hard to test, to be honest. Maybe you could just, you just need very large cohorts with, high quality dietary data, which is a bit difficult, but diet questionnaires, well, actually, it's maybe not as difficult as regular dietary questionnaires because you could really constrain the question to fibers and go, okay, which types of fruits, vegetables, grains do you consume most? And gosh, I mean, that would be a fascinating result. But I think that's part of the feedback to our brains. I think that's pretty interesting.
Yeah. Thank question to Thomas. Is that okay?
It's the mash.
So years ago, I was talking to a colleague, and they were saying that they had a student who used to test their own microbiota in vitro. I believe it was with prebiotic of some sort, I can't remember which. And they were saying that the prebiotic used to work really well. This person was single, lived alone, didn't eat great. And then they started dating someone and they started eating a healthier diet, and they found that the prebiotic didn't work as well because their gut microbiota in general was healthier. So I was wondering if you've ever come across anything like that with people changing their diets. And then as a sort of second question, I was wondering if you've ever looked at, because I'm interested, I'm kind of interested in antibiotics. I'm interested to look at somebody who's been on antibiotics, and then, so you have maybe a pre-antibiotic sample, a sample while they're on antibiotics, and maybe a sample while after they've recovered from antibiotics, and kind of see how their fermenting ability changes before and after. Maybe they haven't recovered fully, or maybe they have an alternative steady state. I don't know if you've ever come across anything like that.
No,
when you had a participant who accidentally went on antibiotics.
They're both great questions. I haven't looked at either of those, but I think that the antibiotics question would be very interesting to look at, because actually I've tended to, you know, especially when it comes to ethics committee protocols and things, just been like, oh, well, let's exclude, as everyone in the microbiome does, you know, anyone who's taken antibiotics, let's get out of there. And the truth is that antibiotics, while especially broad spectrum, are pretty damaging, that, you know, there's really only a fraction of the microbiome that's responsible for fermenting these fibers. And it's unclear to what extent they're damaged by this. So I would be really interested to see. As I say, I assume that you take antibiotics that sit slate clean and then you're recolonized, but I don't think it's that simple.
No, well, at least in my in vitro models, you see some bacteria bloom. So in my case, I was looking at clindamycin, and there's lots of Bacteroides that are resistant, not all of them, but Bacteroides in general just have more antibiotic resistance mechanisms compared to some other bacteria. But yeah, I think people do consider it to be sterile, but you're not really.
Yeah, no, that's a really interesting question. You know, I think this is something that we should actually actively study, because ultimately antibiotics is something that we can't really do without, at least at the moment. And people take them, and just saying, oh, well, you have to retest and nothing at all, you know, no holds barred now. It's a bit cheap. I think there's some interesting science to be done there. So it's a great question.
Thank you, Julie. So another question to you, Julie, from media, and she's asking, she's first saying it was a great presentation, and you mentioned valerite suppressed C. diff infection. What valerite associate with, does valerite associate with bile acids? Some bile acids known to induce C. diff infection. And just to note, you know, from our talk yesterday, I don't know if you attended, but in our work, we have, together with Tom Wolmansky, we have noticed that bile acid had various different effects, differing association with gut microbiome diversity. Some of them were positively associated with it, some of them were negatively. So yeah, this question is about what you've experienced.
Yeah, so we looked also at bile acid. I didn't present the data because it would have been a really long talk, but so with bile acids, we have a better idea, I think, of a better idea of what members of the gut microbiota are involved in all the steps. We don't know exactly every bacteria, but we know some bacteria, lots of Bacteroides, for example, produce this enzyme what's it called? Biosyl hydrolase, space out. And we know some Clostridium species can produce 7-alpha dehydroxylase. These are some of the enzymes that convert deconjugate primary bile acids and convert primary bile acids to secondary bile acids. And the conjugated primary bile acid taurocholate does stimulate C. difficile growth and germination. But if you cleave off that taurine molecule, you get cholate, which is a more weak germinant. And then if you do the secondary bile acid synthesis from there, you get deoxycholate, which inhibits C. difficile. So in that sense, we have an idea, and that's something I worked on with someone named Benjamin Molish at Imperial College, and he did more work into detail on that with C. difficile. With valerate, people, as you know, we've seen focus a lot on acetate appropriate butyrate, which is great, but then we start to have questions, well, what bacteria are producing valerate? And there's some indications out there that valerate is associated with the high diversity of the microbiota. We think that some members, I think it's Silobacter, there's some member of the microbiota that has been suggested to produce valerate, but we haven't really gone through and teased out exactly which members of the gut microbiota are responsible for producing, and that's something that we will have to go through and do eventually, screen a lot of bacteria. But at the moment, that's not something that I'm working on currently. So this is the work I presented today was a lot of work from my postdoc, and I'm now starting my own lab. So I'm looking at a bit of a different idea of looking at the gut microbiota and antimicrobial resistance, but I thought I'd present my published work first. So yeah, so it's something that we're working on teasing out in terms of valerate.
Thank you. Thomas, as a question from Moritz Weber, have you considered testing a particular person's stool with your system to identify diet components that would favor stability or engulfment of particular probiotics believed to be beneficial to that person, maybe in a model-guided fashion in order to reduce the search space for metabolites?
Yeah, I mean, I had not considered that. That's a really interesting question. I think that what I would say is that we're not going to be able to necessarily say anything about engraftment. However, what you could say is that you could demonstrate that, say, if you have a particular probiotic and you identify which fibers are well fermented and used as a carbon source by that probiotic in vitro, then you can provide some evidence that you'll be able to increase its relative abundance transiently. Then when it comes to the mechanisms behind engraftment, that I'm much less qualified to discuss. And I think that ultimately that comes down to some other variables as well. But assuming a mass action effect, then I think that it's perfectly reasonable to say that, yeah, we could use that framework to at least increase our chances. I think that's a very interesting point.
Julie, a question from Sean. So FMT dislodges an established C. diff infection, but many people are colonized by C. diff and carried asymptomatically. Have you explored colonization resistance in your chemostat system?
So like in a healthy gut microbiota, no, we haven't really looked into that too much because we were interested in looking at the effects of FMT, but at least from what we know about C. difficile and other pathogens, we wouldn't expect it to be growing at very high levels if it was present. And we don't consider C. difficile to be present at high levels, even in people who are asymptomatic carriers. Even in people who are on, we know have C. diff infection who are on antibiotics, C. difficile is not necessarily present at extremely high abundances. So yeah, so it's not something not a lot of researchers I don't think have looked into it because I think maybe it doesn't sound as exciting because it's not producing a disease. But yeah, I mean, I think another thing people forget about is that babies often have C. difficile in their guts, but they don't see disease in babies. But it's something that'd be interesting to look at, but not something I looked at personally.
Okay, we're at time. So I would like to conclude this Q&A session and thank both of our speakers for fascinating talks and really insightful Q&A session. So we'll see you back at 12:45 with session number 3 that will focus on in vivo interventions. Thank you again.
Thank you very much.